Okay, so I have the lovely task of trying to make cell signaling, gene expression, lots of abbreviations, things like that, digestible and understandable to people who've not already taken college courses in cell biology and all this kind of stuff. Because I don't want to be up here for 30 minutes talking over everyone's head, and then you're like, so he looked smart, I guess. So I want to make this stuff understandable. So there are going to be some simplifications here. But the general ideas should prove to be useful as we move forward to the bigger picture topics towards the end of the talk. So we talk a lot about stress, recovery, and adaptation at our seminars. And we just assume, or we tell it to our attendees as a given, that this is how things happen. And we talk about specificity, as Sully talked about. And we don't ever really just get into the weeds as to how we get that specific adaptation. It's just kind of like assumed that, like, yeah, we use the suntan analogy. You lay down, you get tan on your back, right? Everybody can understand that. And that's kind of the way that we explain to people how specificity works. But I'm going to explain actually how specificity works here. And this is where this gets interesting, because what actually happens after I deliver the stress to you? How does your body sense the disruption of homeostasis? Because it has to sense what is the stressor that I just gave you. It has to turn that disruption into a signal that the cells can interpret and then act upon to adapt. So there's a bunch of intermediate steps that have to occur that we don't always get into the weeds in explaining. And that's what I'm going to do today, is how this specific adaptation happens, how cells recognize what we're doing to them. And so at the level of skeletal muscle, there's a bunch of different adaptations that can happen. And we traditionally kind of define these or separate them into the structural adaptations, where the muscle proteins, you accumulate more muscle proteins, you build more sarcomeres, satellite cells get into play, and these other ancillary tissues, your connective tissues, nerves, tendons, all adapt as well. But on the conditioning side of things, the more endurance side of things, we talk about the metabolic adaptation. We talk about the biochemistry that happens in the cells that generates all the continuous energy that you might need to do an endurance training bout. So we have these two different adaptations, and that's what Sully got us started thinking about. And so in order to get these divergent adaptations, as he said, we have specific genetic programs that we have to execute in order to get that adaptation. One genetic program will take you down the strength hypertrophy kind of adaptation road. The other program will take you to a metabolic or oxidative adaptation. Or you can actually have atrophy programs. So interestingly, atrophy is not just, we talk about the lack of stress being itself a stressor which you adapt to, and that's in fact true, because you adapt to it by executing atrophy programs. It's not just muscle goes away, muscle actively decides, I'm not needed here, I'm going to go away. So that's how atrophy works, and I'll get into that a little more. And all these pathways obviously interact with each other. We talk a lot about how bone is a living tissue constantly remodeling itself, and that's how it gets stronger when you strength train. Well, your muscles are kind of doing the same thing. They're building up, getting broken down. We talk about muscle protein synthesis, muscle protein breakdown. It's a dynamic state all the time. And when we train, we're just tilting that balance, hopefully, a little further in the muscle protein synthesis, hypertrophy, strength, or in the case of endurance aerobic training, down the metabolic pathway. So just a primer before we get into the genes, I have to remind everyone how genes work. So this is kind of that cell biology basics. So this is what we call the central dogma of molecular biology, where DNA is kind of the cookbook in your cells. It gets transcribed into RNA. It's like if you were to copy a recipe down onto a sheet of paper to go use, you make RNA out of DNA. And then you go and you translate that RNA, the transcript, what you copied out of the book, into the final product, which is the protein, and that's translation. So DNA to RNA to protein is the standard path that we kind of use as an oversimplification, as you'll see. So you guys, everyone in this room, I hope, has 23 chromosomes. About 20,000 genes on those chromosomes are used to express these proteins. The rest traditionally were thought of as junk, but we're realizing that we weren't as smart as we thought, and we're slowly figuring out what those other genes are for. And the whole process is highly regulated, because your body can't handle trying to express all 20,000 genes at once. You don't have the resources to do that. So there has to be a way for your body to decide, what genes am I going to express right now, or which ones can I wait and use a little bit later, or which ones aren't necessary at all right now, right? So that's what goes on here. So you can see down your left side of the screen, DNA becomes RNA becomes protein. Transcription is the first step, translation is the second step. But you can see these extra things that are listed adjacent to it are all the steps of regulation. So at the top you see epigenetic modification. That's the things that tell your DNA this gene is turned off for now, putting the mute button on it, or this gene is going to be expressed. If we're going to express it, we have to transcribe it. Transcription factors help make that happen. Transcription factors themselves can be turned on or off to facilitate that process. Once we have our RNA transcript, you can intervene at that step and regulate it. There are interesting things called microRNAs that interfere with this process. They come in and they can actually block the expression of that RNA transcript. They take your recipe that you copied out of the book and throw it in the trash. Say, no, you're not going to make that right now. Okay? And interestingly, when we talk about anabolic resistance, as folks get older, or sometimes when you might have heard about people who are, quote, non-responders to training, which we have a feeling is not actually a thing, we think that that process of anabolic resistance or apparent training non-responsiveness to a degree might be mediated by these microRNAs that come in and interfere with your ability to express the proteins that you need to adapt. So they've measured some of these things in older people or anabolically resistant people, and they have sky-high levels of some of these known microRNAs that interfere with your ability to express certain proteins. You then take that transcript and you translate it, and so the amount of translational machinery that you have available, like the equipment you have in your kitchen to make the recipe, determines how quick you can crank out this final protein, right? So that's your translational capacity, which itself is regulated. How much machinery am I going to build so that I can then make my proteins? And then after you make the protein itself is then modified to help make it more functional or even potentially degraded. And so when you do an individual exercise session, we always talk about exercise versus training. You get this quick transcriptional response. You make a bunch of RNA transcripts. And that's really hard for us to measure really consistently in the literature, especially when we're comparing all these different types of training programs and modalities and things like that. Instead, when we have repeated exercise bouts, what we know as training, over time those bouts of RNA transcription get turned into proteins, and proteins are much easier to measure in terms of the functional adaptation that you get out of it, because proteins last longer than RNA does. RNA is your recipe, you do it, throw it in the trash, but you have the final result there, and we can measure that. So I like this quote from this review paper that said, the net effect is to promote optimal performance during a future exercise challenge. This results in a robust defense of homeostasis in the face of metabolic perturbation and consequently enhanced resistance to fatigue. Obviously, this sentence is written more with an endurance type bout in mind than what we would talk about, because they're talking about metabolic perturbation, but you could apply the same concept to a force perturbation in terms of when we require high force demands on you in resistance training. So you'll see this diagram that I took, and I pointed out that they claimed that this is a simplified overview of signaling network, and this is particularly as it pertains to the aerobic endurance side of the spectrum. Believe it or not, there is a second beautifully simplified overview for the strength and hypertrophy programs. So this looks super scary. I wish it projected a little bit better, because I included this diagram a few times throughout this to kind of show where we are on here. But hopefully, if you can see, maybe if you're a little closer, you can see the things inside the cell that stand out. The big letters in the middle there is something that's called PGC1A. Okay, so we'll keep this abbreviation in mind. So we're going to start there. We're going to talk about what this PGC1A is and why it was written in big, bold font in the middle of the cell with everything else converging on it. Why does this matter? First off, you can see why we call it PGC1A, because it actually stands for peroxisome proliferator activated receptor gamma coactivator 1 alpha. Fuck that. Okay? It is a protein complex. So it's a protein that's in the cell, and it's considered a metabolic checkpoint. Okay, so again, we're in the endurance, metabolism, the oxidative kind of world right now in terms of our genetic programs. It takes a bunch of signals that are going on, right, and it kind of integrates those and decides what to do with them. So remember, our cells have to sense the stress that they're receiving and turn it into whatever adaptive response we're going to get. So this protein senses what's going on in the cell, what kind of stress am I receiving? And so since we're in the oxidative realm, this is going to be the protein checkpoint that senses when we're doing something like an endurance bout. Okay? So it senses our energy status, our oxidative, reductive, or redox status, and it senses endurance type muscular activity in ways that I'll get into in a little bit. So it is a transcriptional activator. It increases gene transcription, and if you'll recall, gene transcription is when you turn DNA into RNA. So it works together with a bunch of those transcription factors to promote expression of genes in this genetic program. So we have this protein that senses what type of activity am I doing. I need to execute this program in order to adapt to that stress. Should make good sense. And so the actual genes that it expresses end up resulting in making new mitochondria. It's this mitochondrial biogenesis that you see here. And then slow twitch fiber type switching. You execute a program that, let's say you're a lucky individual who is super high genetically programmed for power. You have a bunch of type 2A, 2X type muscle fibers, like my wife, for example, and then you put her and you make her run a bunch of marathons. If you do that for long enough, you can get some of this fiber type switching where you turn into a more of a type 1 type profile. For most of the people in this room, it's probably not what you guys are interested in, but some people are into that kind of thing. So when you have acute exercise bout, the activity of this protein goes up. If you keep training with this endurance style modality, you get increased expression of that protein. So there's more of it around all the time doing its thing. So now in comparison, we have our other simplified overview on the strength and resistance training end of the spectrum. And you'll see a similar big central protein right there that you see, mTORC1. Okay? That's kind of where your eyes should be drawn in this. Why is that so big and bold? That itself is another protein complex called mammalian target of rapamycin complex 1, or mTOR, as we're going to call it. And that's going to be the main anabolic checkpoint. It's kind of the analog for PGC1 alpha, except over in the resistance training realm. This is the protein that will integrate more of these signals. What kind of stress am I receiving right now? What kind of demands are there on me as a muscle cell? And turn it into a plan to execute a particular genetic program. That genetic program will be, as you see down below, protein synthesis. Okay? That's that MPS that Jordan likes to talk about all the time, right? So mTOR, again, integrates these signals like nutritional status. Leucine, that's why we talk about leucine all the time. Insulin, insulin-like growth factor. And then mechanical stress. Mechanical stress like having a 405 bar on your back, right? And so this protein actually acts on the translation part of the pathway. So if you recall, PGC1 alpha was a transcriptional activator. It activated transcription where you take DNA to RNA. mTOR instead acts on the translation side of things when you take your RNA transcripts and turn them into protein. So it acts on a different part of the pathway. And that's muscle protein synthesis, which is itself translation. So we have a bunch of primary stimuli that our cells sense. These are all the things that our cells experience when we're exercising. And they want to turn these stresses into an adaptive response. This is where stuff gets interesting in terms of how you can explain, to a degree, outside of the paradox that Sully was talking about, how adaptive specificity happens. So when I exert mechanical stress on you, when I make you pull 500 pounds off the floor, there's a lot of interactions between the cells, the tissues, and the proteins within your muscle cells that sense the tension, that sense the mechanical stress. They sense that I'm holding 405 pounds in my hand, okay? And they can turn that stress into a signal that's called mechanotransduction. Fancy word. Transduction just means you're taking one signal and converting it into another type of signal. So it's taking the signal of the mechanical stress, the weight, and turning it into a cellular signal, that's the mTOR, that then goes and makes you synthesize muscle protein. So mechanotransduction, again, since it doesn't project well, it's not worth me pointing out here, but the font's too small anyway, no big deal. Mechanical stress is one of the things that, if you'll trust me, has an arrow pointing towards mTOR there, okay? Mechanical stress also results in muscle damage. A bunch of these cellular structures that Sully talked about, the sarcolemma, the T-tubules, things like that, they get sheared and damaged acutely when you pull a really heavy weight off the floor. And that generates local acute inflammation. You get some muscle inflammation. Cytokines, hormones, a bunch of growth factors are secreted locally right around that muscle cell. And those end up having kind of effects to help promote the recovery and the adaptive response. They recruit satellite cells, which are kind of neighboring cells around the muscle cells, that can come in and donate and give extra nuclei to the muscle cell. So it has more genetic material, because genetic material, DNA, is what's inside nuclei. So you have more nuclei that you can then transcribe more from and translate more from and get more muscle protein. And it also inhibits myostatin. Myostatin is an interesting protein that's not super well understood, but it's basically the protein that ends up kind of holding the reins back so that your muscles don't get super jacked out of control. It actually is. So there are these things you can Google. Yes, you can Google myostatin knockout, like Knockout, and see some fascinating pictures of creatures that don't express this protein, just absurdly jacked creatures. Jillian Ward. Yeah. So it's very interesting. So that local acute inflammatory response when you train actually blocks that from happening. So it kind of says, all right, I'm clear to get bigger and get stronger right now. You don't need to hold me back, okay? So calcium is the next one, and that's what Sully mentioned, because calcium is what actually, the floodgates open, calcium leaks out into the muscle cell, and that's what activates muscle contraction. So you can see here is a useful way for a cell to sense, oh, I'm contracting right now. I need to adapt to that contraction. So there are these proteins called calmodulin and calcineurin, and they actually are able to sense the calcium as it comes into the cell, because every contraction, calcium leaks into the cell, and then after the contraction's over, it gets packed back away again. Calcium leaks out for the next contraction, it gets packed back away again. So the calcium is kind of oscillating back and forth in the cell. So these proteins sense how this calcium is coming into the cell. When you have a really brief, high power output contraction, you get really transient, really brief, but super high spike in how much calcium is coming out. Those proteins can sense that and say, oh, I just saw a massive amount of calcium, but it was like for a split second. In comparison, you run your 10K, you need that repeated cycling, so the calcium is constantly oscillating back and forth, and on average, there's probably more of a consistent level of calcium in the cell. Of course, it's just getting packed away and coming out more frequently, but that can be sensed in its own way too. So you have more steady currents that get sensed themselves. So this is a way for the cell to exercise what's called excitation-transcription coupling. If you think about what these words mean, the muscle cell gets excited, not in like the literal sense, but in like the depolarization sense, and it contracts. Calcium comes out, those proteins sense how much calcium came out, and it can turn that signal into a transcriptional or a translational response. Can turn your aerobic activity, where you have a steady calcium current, into a PGC1 alpha response to go down the aerobic metabolic adaptation pathway. If you have really high calcium spikes, it can turn that down into an mTOR muscle protein synthesis response. ATP turnover is another pathway, just because of the different energy demands from these two different ways of exercising, basically. When you have really consistent activity, when you run your marathon, you are constantly burning through energy. Really, really, really high amounts of energy, really consistently, right? Compare that to your 1RM, compare that to a PR snatch. It's done in a split second. You used a lot of ATP, but really quickly, and then it gets recycled by your creatine system, and you're good to go. And you can go sit down and scroll through your phone until you do your next rep 10 minutes later, right? When you're running the marathon, you're not stopping. The energy is consistently being churned through, and the cell has a way to sense that. It has a way to sense that signal of, oh, I'm low on energy constantly right now, versus your PR snatch, I'm good to go on energy. My creatine helped me out after that rep, okay? And that's done through this protein called AMPK, or AMP kinase. So that results in an intensity-dependent activation, okay? And so this then, this AMP kinase protein inhibits, so when AMP kinase is turned on, it basically means that it has sensed that your cells are in a low energy state. Because when you use ATP, you make ADP, AMP. So if you use up ATP, you use up all the energy while you're running, you might get some AMP accumulating. That turns on AMP kinase. That then blocks anabolic pathways. Should make sense, right? You don't need to be getting jacked during the marathon. You need to be worried about having enough energy to finish the marathon. So that's what AMP kinase does. It restores your cellular energy status. So it tells your cells, I need to break down glucose, I need to break down fat, I need to make more energy so that I can make ATP and have enough energy to finish this race, and we don't need to do muscle protein synthesis right now because we have a more important task we're doing right now. So it senses the stress of having low cellular energy and turns that into this response of, I need to get more cellular energy, right? That robust defense of homeostasis that we talked about. When AMP kinase is turned on all the time, it actually ends up activating this PGC1 alpha thing that we keep coming back to, because your muscle cells are getting the signal all the time that they're low on energy. When you practice your 10K every day, your cells are like, I just can't make enough energy to keep up with this demand. So I'm going to turn on PGC1 alpha. I'm going to make a bunch of new mitochondria so that I can make more ATP. I'm going to switch to a slow twitch or type 1 muscle fiber type that can handle these efforts better. Does that make sense? That's how you're generating that specific adaptation to the task. Finally, hypoxia is another one. So when you're at rest, the muscle cells perceived the kind of amount of oxygen that's swimming around it is about 1/5 the pressure of oxygen in the air that we're inhaling right now. When you exercise acutely, that concentration goes way down. You're churning through it a bunch more. When you have that acute hypoxia during the exercise bout, again, you can imagine this is more often the case when you're doing an endurance task, this protein called HIF1 gets activated, hypoxia-induced factor. And that then goes on, so that's basically sensing the signal of low oxygen, and then it's going and activating gene transcription. It's making you make more proteins according to the stress that it is perceiving. So erythropoiesis is a fancy way of making more red blood cells, right? That's why people like me when I swam, or other types of athletes, they'll go up and swim at altitude, or they'll run at altitude or whatever, so that oxygen is even lower. This protein's turned on even more. You make a bunch of new red blood cells. Or you can just blood dope instead, right? Angiogenesis, make more blood vessels to deliver that blood more effectively to the muscles. We'll talk about this in a minute. And then increasing your methods to execute glycolysis and energy metabolism so that you can make more ATP to handle the energy demands better. So this results in better oxygen delivery, better oxygen extraction from the blood, and improved, that should say, aerobic metabolism, not anaerobic metabolism. Happens. And finally, I want to talk about atrophy, because as I mentioned, atrophy is its own genetic program. It's not just muscles passively going away. It integrates upstream signals. So I don't know if anybody can see on the next picture here, adjacent to the mTOR that you see there, you see this, FOXO right here. Stands for something not super important. But FOXO, just like we had the anabolic checkpoint of mTOR, or we had the metabolic checkpoint of PGC1 alpha, FOXO, this 1 and 3, are the atrophy checkpoints. You can see the pattern the same way. They integrate a bunch of signals that they're getting, and they turn it into a response. So the upstream signals might be mechanical unloading, right? Like sitting on your ass all the time, going up into space, being sick and bedbound for a long time. Chronic hypoxia or chronic inflammation. This is why I see patients who have bad COPD, or they have bad autoimmune diseases or something like that, and they tend to not be super jacked, believe it or not. Myostatin is another signal that has a role here. Corticosteroids, like if you have clients who happen to take prednisone more than a couple days, it takes a little while longer, but that actually influences this pathway. And so this FOXO gene senses all these things, and it turns it into a response. It activates these downstream atrogenes. I don't know who decided that was a good word for it, but atrophy genes, okay? So genes that inhibit anabolic processes, prevent you from making new muscle protein synthesis products, and basically activates your way of making your muscle cell mass, your total muscle mass, actually decrease. So that's the adaptive response to all these things. So in summary, you can see how these primary stimuli of how your muscles sense mechanical stress and transduce it into a response how your cells sense the calcium oscillations that occur with every contraction and transduce it into a response, how they can sense how much energy they have available, turn it into a response, again, in accordance with the task at hand, or how they can sense hypoxia, which is less relevant for strength training, but more relevant for conditioning activities. There's also a few others that, for the purposes of simplification and the fact that it would require a whole bunch more background explanation, I didn't get into, like reactive oxygen species and things like that. So I left that one out. Finally, I'll touch on the interference effect. CJ is going to talk about the implications of this a bunch more. But we all kind of intuitively have an idea that when you try to do both of these tasks at the same time, you tend to not get the greatest results on either end, right? So there's something going on that affects your ability to simultaneously get very, 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 very conditioned and very, 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 very strong. Because these are divergent stressors, you tend to get these divergent adaptations. But Sully touched on this with the high intensity piece, and how you can have this kind of paradoxical response where you have an exercise modality that leans toward one end of the spectrum, but you're also getting really good adaptations at the other end, which suggests that there's this spectrum. It's not just a complete on-off, I'm pure muscle protein synthesis right now, or I'm pure aerobic adaptation right now. There's a spectrum, there's a balance, and again, this is all in constant dynamic flux, right? I think I'm reasonably strong right now, but I could still go outside and run a 5K around this neighborhood and produce an aerobic metabolic adaptive response. There's a spectrum. It's all the time. So how might this happen? I think there's probably a number of different ways that this happens, but the one that is currently thought to be one of the most influential is, if you'll see here, can you point to AKT over there? So right before the step where you get down to expressing mTOR, there's a little guy coming in and blocking that from happening. And it might be a familiar abbreviation called AMPK. So you can, if you'll recall, I mentioned AMP kinase is the protein that senses your cellular energy status. So when you run the marathon, your cellular energy status is real low. And so it blocks activation of mTOR. Because it says, again, we don't need to be synthesizing a bunch of new muscle protein right now. We need to be worried about maintaining our cellular energy status. So that's one theory, particularly with chronic training, you end up expressing AMP kinase a bunch more, and that might interfere with the expression or the activation of mTOR and subsequent muscle protein synthesis. So that's one potential mechanism for how this interference effect between endurance training and resistance training might happen. When you're low on energy all the time, your cells are not worried about making a bunch of new muscle protein. And so the other piece that I was going to touch on was tissue level adaptation. And I'm just going to be real brief here because you guys are familiar with the structural adaptations that happen when somebody resistance trains. Muscles get bigger, you get more protein, your tendons get thicker, stronger, your bones get stronger. And when you do the endurance activities, as we've expressed, you mainly upregulate metabolic or biochemical processes, less so structural processes. So these are the kind of things that change in your skeletal muscle from a structural standpoint, particularly when you do resistance training. We already know this. It's not that interesting for me to go into right now, okay? So I'm not really going to. The thing I will mention is this topic called muscle capillarization, okay? So capillaries being the small blood vessels that kind of let the blood that's in your arteries get deep into the muscles, okay? They help to deliver blood into the muscle tissue. And in the literature, capillaries, or this capillarization is measured as capillaries per unit of muscle fiber area. So how many capillaries do I have for a given, for a given amount of muscle mass? And this has interestingly been found to correlate decently with someone's VO2 max. So how many capillaries they have per unit of muscle area. There's a bunch of small studies on this. They aren't super awesome. There's some data that conflicts between them. But as of the data that we have now, what I was able to glean from it suggests that when you do resistance training activities, you tend to have a proportional increase in capillarization, meaning your muscles get bigger, but you maintain the same capillary density because your muscles effectively need the same amount of blood that they got before. So if you have more muscle that is going to need proportionally more blood, you'll produce proportionally more capillaries. That makes kind of intuitive sense, right? On the other hand, when you do more of an endurance training task, For example, you might activate that hypoxia-induced factor, for example, and that factor tends to make you want to make more blood cells, make more blood vessels, to make you more efficient at delivering the oxygen. So in that situation, this is actually fairly well described, that you have an increase in capillary density. So for the same amount of muscle mass that you have, because again, you're not dramatically increasing how much muscle mass you have when you do one of these high endurance tasks, for the same amount of muscle mass, you have way more capillaries there. That makes you way better able to deliver oxygen to that tissue. Does that make sense? So this is kind of an interesting thing to point out, because from the standpoint of muscle capillarity or capillarization, in this context, it suggests that resistance training does not impair aerobic or endurance type performance. Anyone see why that would be? Because you have the same capillary density you have before. So that's not the limiting deal here. That means that you do your resistance training tasks, your muscles get bigger and stronger. You don't automatically get worse at endurance training as a result of that because you don't have enough capillaries anymore, because your ratio is still the same. Proportionally, you're still delivering the same amount of blood to the tissue. So that's why we don't think that this has anything to do with, that resistance training has anything to do with impairing endurance performance from the tissue level standpoint in terms of capillarity. I think that's all I have from explanation stuff. Hopefully that was clear. And so a few other questions is that really I focused on the main signaling pathways of PGC and mTOR here, but you saw that simplified overview, if that was the simplified overview, you kind of get an idea of how things actually might work, being way more complicated. And so there are a number of mTOR independent pathways for skeletal muscle adaptation. What happens when we train? A bunch of things that happen outside the context of mTOR, like your satellite cells having a role in the process. Ribosomal biogenesis. Ribosomes are the machinery that you use to translate RNA. So might there be a response in terms of us building a bunch more ribosomes so that we can more effectively translate these RNAs and do muscle protein synthesis? So there's a bunch of other pathways that haven't been as well described. And we know they have a role, just the relative contributions of their roles remains to be clarified. The role, mechanisms, and interactions of post-transcriptional regulation. You remember I mentioned those microRNAs that probably have a role in anabolic resistance and a few other processes in terms of variable responsiveness to training. Why does one person get insanely good after training and another person doesn't? Well, everyone in here is probably just like, yeah, it's genetics. Well, this is the genetics that I'm talking about, right? This is that stuff. So we need to understand that a little bit better. And then figuring out a little better the genetic mechanisms of this interference phenomenon. Because I mentioned the AMP kinase one, but that seems a little too simple to me to be able to explain the whole thing. So there's probably a bunch more ways that this happens that we don't know quite enough about yet.